What's up, everyone? Today we're going to go over the major cytogenetic testing methods used in clinical genetics. The goal of this video is not just to memorize the tests, but to really understand what each one is good at, what its limitations are, and when you would see it clinically. Some tests look at large chromosome structure. Others look for very small copy number changes. Some can detect balanced rearrangements, while others completely miss them. And honestly, one of the most important concepts in genetics is no single test detects everything. So let's build this step by step. Now let's look at the big picture. Historically, chromosome analysis evolved from unbanded chromosomes to banded karyotypes, then FISH, then microarray technologies, and now next-generation sequencing approaches. The overall goal of all of these technologies is genotype or karyotype to phenotype correlation and helping determine recurrence risk. But each technology has distinct strengths and weaknesses, which is why choosing the right test depends on the clinical question being asked. Let's start with conventional cytogenetics, the classic karyotype. A karyotype allows us to visualize the overall chromosome structure and number under a microscope. This is especially useful for detecting aneuploidy, large deletions or duplications, balanced translocations, inversions, and marker chromosomes. But resolution is limited. A standard karyotype typically detects abnormalities around 5 to 10 megabases in size. So smaller microdeletions or duplications may be completely missed. And this introduces one of the most important teaching points. Higher resolution does not automatically mean better. Different tests are better at detecting different types of abnormalities. For example, a karyotype can detect balanced rearrangements, but a chromosomal microarray usually cannot. That distinction becomes very important clinically. Next is FISH, or Fluorescence in Situ Hybridization. FISH uses fluorescent DNA probes that bind to specific chromosome regions. One of the easiest ways to think about FISH is as a targeted search. Instead of scanning the entire genome broadly, you're asking, do I see this specific region? And because it's targeted, FISH can be very fast. A major advantage is that interface FISH can be performed without tissue culture, which makes it useful in rapid prenatal diagnosis. FISH is commonly used for rapid prenatal aneuploidy detection, microdeletion syndromes, subtelomeric rearrangements, marker chromosome characterization, and confirmation of array findings. A classic example is 22q11.2 deletion syndrome. Now here's the limitation. FISH only detects what the probe is designed to detect. So if the abnormality lies outside the targeted region, it may be completely missed. And if the deletion is smaller than the probe itself, FISH may not detect it. So FISH is excellent for focused questions, but not for broad genome-wide screening. Now let's move into chromosomal microarray, also called array comparative genomic hybridization, or aCGH. This was a major advancement because it allowed genome-wide detection of copy number changes at much higher resolution than karyotyping. Microarray compares patient DNA to control DNA and looks for gains or losses across the genome. This makes it extremely useful for detecting microdeletions, microduplications, and other submicroscopic copy number variants. This is why chromosomal microarray became first-line testing for many patients with developmental delay, intellectual disability, autism spectrum disorder, and multiple congenital anomalies. But here's the major limitation. Microarray detects dosage imbalance, not balanced rearrangements. So if a patient has a balanced translocation with no gain or loss of genetic material, the array may appear normal. The array can tell you that extra or missing material exists, but it often cannot explain the structural mechanism behind it. That's why follow-up cytogenetic studies are frequently required. For example, is trisomy 21 caused by non-disjunction, or is it due to a Robertsonian translocation? These have very different recurrence risks. Now, within microarrays, probe type matters too. BAC arrays use larger probes, which means they may miss smaller alterations, but are less likely to detect uncertain findings. Oligonucleotide arrays use much smaller probes, giving higher resolution and allowing detection of very small changes. But higher resolution also increases the likelihood of identifying variants of uncertain significance. And this becomes a major counseling consideration. Next is SNP array technology. SNP arrays can detect copy number changes similarly to aCGH, but they also provide genotype information. And this allows them to detect something extremely important: copy-neutral loss of heterozygosity. In other words, the copy number may appear normal, but both chromosome regions came from the same parent. This allows SNP arrays to detect uniparental disomy or UPD, regions of homozygosity, and loss of heterozygosity. This is especially important in imprinting disorders like Prader-Willi and Angelman syndrome. Now, SNP arrays still share some limitations with aCGH. They generally cannot detect balanced rearrangements, and they may not fully explain the mechanism behind a dosage abnormality. Finally, let's talk about next-generation sequencing, or NGS, because this is where modern genetics starts to overlap heavily with cytogenetics. Unlike karyotyping or microarray, which primarily look at chromosome structure or copy number changes, next-generation sequencing is focused on reading the actual DNA sequence itself. And the reason it's called next-generation sequencing is because it allows millions of DNA fragments to be sequenced simultaneously, rather than one fragment at a time like older Sanger sequencing methods. So instead of looking at one gene individually, NGS can analyze panels of genes, the exome, meaning all protein coding regions, or even the entire genome. NGS is strongest for detecting sequence-level variation. So it's excellent for identifying single nucleotide variants, small insertions and deletions, and increasingly certain structural variants depending on the platform and analysis pipeline. But historically, NGS was not as good at detecting large chromosomal rearrangements as traditional cytogenetic methods. And this is where the fields of molecular genetics and cytogenetics have really started to merge. As sequencing technology and bioinformatics improve, NGS is becoming more capable of identifying copy number changes, structural rearrangements, and even chromosomal breakpoints that older technologies might require multiple tests to characterize. One major theme across all these technologies is the importance of parental studies. Parental testing helps determine whether a finding is inherited or de novo, the mechanism behind the abnormality, and recurrence risk. And often, multiple technologies work together. For example, a microarray may identify a duplication, FISH may clarify its chromosomal location, and a karyotype may explain the structural mechanism. So in practice, No single test tells the entire story. So if you take anything away from this video, first, let it be these 3 concepts. 1. Every cytogenetic technology has strengths and limitations. 2. Different tests detect different categories of abnormalities. And 3. Understanding what a test cannot detect is just as important as understanding what it can detect. And that's your review of cytogenetic testing methods. Hit that like and subscribe button for more genetics content, and I'll see you next time.